with that, I would like to introduce our speaker, Professor Aedín Culhane. Aedín is Professor of Cancer Genomics at the University of Limerick School of Medicine. She is also the director of the Limerick Digital Cancer Research Centre, and she is co-lead of the All-Ireland eHealth Hub for Cancer Award. Her lab is leading efforts in genomic open source software development and methods for federated analysis of genomics and healthcare data, and I'm sure we'll hear a lot about that over her lecture. She is a computational biologist and a leader in bioconductor. And she also is, this is a very complicated bio with a lot of accolades. And I think what I'll do is, I think we're going to hear a lot of what she's working on during the presentation. But Professor Culhane actually moved to Ireland in 2021 after spending 15 years at the Data Science Department in the Dana-Farber Cancer Institute and Harvard University in Boston. And we had invited her to speak around the area of cancer data and cancer genomics, with a particular maybe focus on the eHealth Hub that she's leading out on, and the importance of data in our cancer healthcare system. So Aedín, I'll pass over to you, and you're very welcome.
Thanks so much, Roisin. Thank you for the invite, and I'm very, very honoured to open this seminar series. So I was asked to speak about cancer genomics and cancer data, but I think hopefully a more accurate title is Joining Up Cancer Data to Enable Research and Time-Sensitive Care. And I would hope that from this you would see how globally research is trying to develop approaches to actually join up healthcare data so we can actually better treat patients, but also that data is available for research and for research insights. And I'll be talking a little bit about that. Okay. And as for conflicts, I've got no disclosures. And as mentioned in my bio, I'm leading the Limerick Digital Cancer Research Centre in Limerick, which really tries to use data to drive medicine and to drive cancer research. 1 in 2 people in Ireland will be impacted by cancer. And cancer is not just driven by our genetics, but also our somatic mutations. And I'll talk a little bit about that in a second. But this is an accumulation of lifetime exposures that damage our cells. So our tissue is exposed, like our skin is exposed to UV radiation from the sun. Our cells are exposed to chemicals, to viruses, to toxins and pollution, smoking in our environment. And this damage over time, which accumulates as we age, predisposes us to cancer. Now, 1 in 2 in Ireland will be impacted by cancer, and Ireland has one of the highest instances of cancer in the EU. We also have a relatively high mortality. Smoking and alcohol consumption contribute to cancer rates. We know that, but do not solely explain the high rate of cancer in Ireland. It could be that we have better overall population surveillance, or it could be other factors. We don't know, and until we have the data to interrogate that, it would be difficult to identify Irish-specific and cancers or mechanisms or risks. And that's really where we need to bring the data together so we can customize prevention for the Irish population. So one of the important things in cancer is really how it has transformed over, I would say, the past 10, 15 years, and really even over the past 5 years. in the 1980s, 1990s, if a patient presented with cancer, we really had just chemotherapy and radiation as available as therapeutics for that person to treat that disease. Whereas insights from genome sequencing projects have actually revealed more detailed biological understanding of cancer, which have led to genomically indicated medicines. In 2007, and this is data from Memorial Sloan Kettering Institute in the US, when they looked in 2017, so that's 7 years ago, about 7.5% of treatments at that time had a genomic indicator. In 2022, that was up to 21%. And I think we're looking now at about 30%. In the past couple of years, the most medications that have been approved, 80% of medications that have been approved have a genomic indicator. And so it's really, really important for treatment that we actually know the genetic basis of the cancer. Now, when we think about genetics and genomics, we need to separate out what's germline and what's somatic. So germline mutation is a familial or inherited mutation. So that's what is in your ancestry, what you get from your mother or your father, from the egg and the sperm cell. Those mutations will be in every cell in your body. Although there are caveats to that. There are cases where in very early in development, maybe a mutation is introduced, and that can lead to some cells having the mutation, other cells not having the mutation. But by and large, anything that's an inherited mutation comes from the egg or the sperm. Whereas in contrast, somatic mutations are not inherited. Those are mutations that are alterations of the DNA that occur over our lifespan. They tend to be localized to tissue. Most cancers occur in epithelial cells, and epithelial cells are those barrier cells. So the lining of the gut, the ducts, the tissue that connects what's outside and what's inside. And those epithelial cells are exposed to a lot of damaging agents. So our skin is exposed to UV radiation, our lungs are exposed to pollution or to smoking. And those cause damage in the tissue, and that's DNA damage. And as you can expect, as we get older, we accumulate more damage. And that's why aging is one of the biggest risk factors for cancer. Now, what's really important about the somatic mutations is those are localized to a tissue. So if we design a treatment that targets that somatic mutation, it doesn't impact all the other cells in our body. It just can target just that cancer. So targeting somatic mutations has been a very attractive therapeutic strategy because it gives us a very specific way to just attack the cancer. And there are some genes that tend to be predisposed to being involved in cancer. And so somatic mutations in these genes are more frequently mutated. So, as I mentioned, somatic mutations can be sometimes a spontaneous error during replication. Patients that have errors in their DNA repair genes, for example, in BRCA1 and BRCA2, accumulate more somatic mutations in their tissue over their lifespan when they're exposed to DNA damaging agents. Equally, we can incur somatic mutations as a result of environment, to smoking, to UV light. And it's thought that obesity and diet can also damage and cause an inflammation signal, which may damage the DNA. And so in this little blue section here, I've got a list of kind of known environmental agents. I'm just going to move my thing so you can see this there. So like air pollution, there's some really nice work from Charlie Swanton's lab at the Sanger showing this. Contaminants in water, pesticides, cigarettes, plastics, etc. And then the endogenous stresses in our cells that may provide an environment that mutations might accumulate, aging, obesity, stress, for example. Okay, I'm going to move this back. Okay, and this is a really, really nice review, just recently published from Charlie Swanton and others about embracing cancer complexity. I definitely recommend reading this. Okay, the second thing that has really become apparent just very, very recently is how there is such a geographic and temporal impact of these somatic mutations. So there's a really nice study from Paul Brennan's lab, who's in Lyon in France, and they looked at the geographic variation of somatic mutations in kidney cancer. And what we see is that areas where people are exposed to kidney toxins, you see a specific pattern of mutation in their DNA. So in this case here, there was an area along the Danube where there's actually plants that grow along the Danube, which are actually kidney toxins. And they could actually specifically detect that signature in the DNA. Equally, there was a signature that was identified that was specific to Japan. So if you look at this, can you see my mouse? Hopefully. If maybe a thumbs up or something.
Yeah, Aedín. Yeah, we can see
good.
you.
Thanks, Kelly. Cool.
Yeah, no problem.
So if we look here, just in this panel B, you'll see that Japan has that green bar, which is unique to Japan. And that actually is a mutational signature which is only seen in Japan. And as you can see, many, many different countries were included in this study. So the group are now going back to Japan and saying, well, what is it that's causing this specific mutograph in the Japanese population? Is there something in the diet or the environment there that's causing this? And equally, you'll see there that in Romania there and Serbia, there was specific mutations that were very high in that prevalent in those areas. So this is why it's really important within an Irish context that we understand the mutational signatures that are observed in Irish cancers. Are they the same as in Western Europe, or are they different? Is there anything that's different in what we observe in cancers here? To date, we don't know this because these aren't studies that are being done in Ireland. So if we look at US data, where most of this data is coming from, like Dana-Farber and Memorial Sloan Kettering were some of the comprehensive cancer centers to actually take on sequencing of every single cancer. So in the Farber, every patient presented was offered sequencing. And that has led to a generation of large databases, which then allow us to identify the genes that are more frequently mutated. And these have led to drug discovery in this area. So where we've identified mutations that come more frequently in certain cancer types, then we can design drugs that actually target those. And again, when you're somatic mutation, you're going to target the cancer and not necessarily the normal cells because it's a specific damage in that tissue. And these have led to the basis of the Foundation 1 and the TSO500 genes. So these are genes that we know that are more frequently mutated in cancer that are now actionable genes. Okay, so actionable means that these direct care. So either there is a precision oncology medicine that specifically targets that mutation, for example, BRAF V600E in melanoma, or we know that that mutation can predispose you to more aggressive cancer or to sensitivity to one or more drugs. Now, but when we look at these databases, OncoKDP at Memorial Sloan Kettering and Molecular Oncology Almanac, Dana-Farber, they only contain drugs that are approved by the FDA in the US. there actually is no database at the moment which is designed specifically for European Medicines Agency approvals. And we have looked for those. So when we realized this, we said, well, actually, let's take the data, let's put it in a machine readable format so we can actually do reporting that's specific for EMA recommendations. And also let's do this for Irish recommendations as well. So when clinical reports are actually generated, these are targeted and customized for European and Irish patients. And within the Irish context, where the public patients tend to get the NCCP approved drugs and private patients have access to most all drugs approved by the EMA, there may well be a difference there. So we sought to understand that difference more clearly. So this is work with Brendan Reardon, who's a PhD student in my lab, but is also a software engineer in the Val Allen lab at Dana-Farber. So he's actually based in Boston, but is doing a PhD with me. So what Brendan did, Brendan curates the Molecular Oncology Almanac database at Dana-Farber. So he has a lot of expertise in curating these indications, and he's very, very knowledgeable on that. So what we did is we went through the EMA databases, and thereafter we pulled the PDFs from the NCCP website, and then we have worked, and the NCCP have been very generous in their time with us and have helped us ensure that our database is accurate and captures all of the different indications that are approved. Okay, we now have a very, at the moment it's still a rough database up on the website, but that will actually be fully searchable and indexable hopefully before Christmas. So the Molecular Oncology Almanac is actually, if you go to molecularoncologyalmanac.com, that will give you the FDA indications. And if you go to IE, Molecular Oncology Almanac, it will now give you the Irish and the EMA indications. At the moment, it's just a big long list, and we haven't actually put up the full database and it's not searchable yet. We are just finalizing the final details on that, but it should be available before Christmas. Why is it important to have this database? Well, this can actually enable clinical reporting. So for patients that have genome sequencing, and then we want to identify of the mutations that are, sorry, let me just maybe go back one. So within these databases, each gene drug indication is listed by which cancers they're approved for, what was the evidence. So in this case, it's all of the NCC approved and reimbursed medications, and then what the therapy is. And this is fully searchable. It is also available within a pipeline for reporting. The pipeline for reporting is in actually consistent with the ESMO guidelines. So both Brendan and Ellie were contributed to the recent ESMO recommendations for clinical reporting. And so the new Irish database that's going to be available hopefully before Christmas will also be consistent within that pipeline to generate ESMO compliant reports. And this is kind of the example of what these types of reports are. All indications are by tier 1, tier 2, tier 3, where tier 1 would be approved medication. And then obviously once you get into, there's a clinical trial available, or there's biological evidence for these, then we can begin to pull from the US databases at that point in time. Because if there's biomarker or preclinical or clinical evidence that's not yet got into a clinical trial, that's going to be the same globally. We haven't yet, and we will do sort of, probably an additional tier here. So it's like, it's approved by NCCP, it's approved by EMA, by the European Medicines Agency. There's a clinical trial anywhere, and there's a clinical trial in Ireland, and then there's preclinical evidence, and there's biomarker evidence. So we'll probably have those tiers, but that's going to take a little bit of work. So hopefully this is useful. So then when we look into the actual medicines that are approved, and we recently presented this at ESMO, but we have, and we're preparing kind of the reports and all of this at the moment, we see that of the drugs that are approved, that these are just the genomically indicated precision oncology drugs or that have a genomic biomarker that's an indication for prescribing that medication. Of those that are approved by the European Medicine Agency, about 65% of those are approved within the NCCP system. And what's happened is the number of drugs that are genomically indicated has risen really exponentially in the past couple of years. 4 or 5 years ago, you maybe had 1 drug per year, whereas now you have 20, 30 drugs. So it's a lot more, and these are taking longer and longer to approve. So we really need to think of ways that we can prioritize these drugs. So one thing we looked at was the overlap by cancer type. We've also looked by gene and a couple of other ways as well. And we're trying to think of what might be a good way to think about, ways to rank those genes of please, these are the drugs that are going to be really important. So if we look across cancer types here, we see the green here is approved by the HSE. The orange is by both, and blue here is by EMA. Now, this was an earlier figure. This is slightly changed from the numbers that we have at the moment, because there was some hospital-approved medications that weren't in this original figure that's here. But you can see that in some cases, for example, here, in endometrial cancer, we have drugs that have been approved by the EMA, but there isn't actually a drug available. This is a genomically indicated drug. This isn't chemo, this is genomically indicated drugs available within the Irish system. Whereas in other cases, we have a lot of drugs that are available in both. So this is kind of my first little section of kind of where we're really trying to take the informatics and have clinical impact. The second thing that when I moved from Boston to Ireland, one of the things I was really eager, and I think I've probably spoken to a couple of people on the call here, was how we actually get patients into clinical trials. And so in order to get patients into clinical trials, it's really, really important that their data is accessible on a national scale, so that we can actually build an accrual plan of how many patients might be on this trial. And that means we need to be able to share data within and between institutes. And recognizing the challenges in this area, quite soon after coming back, myself and Mark Lawler put in for a grant to the HEA Shared Island North-South Research Programme, and were successful in getting funding for an emerging hub of research excellence, which is a significant grant. And that has really allowed us to try to both do more detailed landscape of what is the data infrastructure at different clinical sites, but also then develop ways that we may be able to assist and develop research in this area. Because I think we can do a lot of phenomenal research in the academic side of things, but unless that's met also at the clinical side, we're never going to impact patients. So it's really important to me that the academic research and the clinical research are both growing and are both supported. So towards that, there's a couple of different initiatives that we're engaged in the lab towards federated sharing of clinical data. One is the Genomic Data Infrastructure Programme, and this is an Elixir program to share whole genome sequencing and other types of sequencing data across the EU. This project is primarily designed for the 1 Million Genomes Programme, which will share the genomes of Europe, but also has cancer use cases. Currently, Ireland is an onboarding nation, and we're working to develop the infrastructure, and that it expands. We're doing a lot of the technical side of it, but other groups, this is being led by Jean-Pierre Cavalera's group in Dublin, and they're doing a lot of the really hard work on the legal side of things that also needs to be done. Okay, but towards sharing cancer data in and for these genome sequencing projects, and we needed to define the data dictionary of clinical variables that were going to be essential. Now, there's a couple of these publications that have come out recently. We are but one, but actually they're all quite similarly aligned because there's overlapping researchers on these. So this is the 1 million genomes, minimal data set for cancer. It contains these variables here. And it's very closely aligned with the IQVIA MEDONC minimal data set, should you have heard of that one. And I can talk about this in more detail should people and want to know, but just to sort of say that, you know, whereas in some of the big cancer hospitals in the US, the movement is towards nearly sharing larger amounts, so we can begin to look at kind of adverse effects and comorbidities more broadly. Within the European context, where we have GDPR, we're very much focused on sharing minimal information, and these were what was considered to be the essential minimum information available. Okay. Of course, within the Irish, sorry, I didn't mean to. I want to trying to move myself here. I'm just moving out there. Of course, within the Irish context, we may think of sharing these minimal data sets, but first of all, we actually have to get this data. As many people on this call may recognize, we don't have an EHR system, electronic health record system, that's nationally implemented. So one of the first things we're actually doing is just working out how each of these variables is actually available at each of the different clinical sites. And I'll explain a little bit more about how we're doing that. And so the eHealth Hub for Cancer is an all-Ireland program led by the University of Limerick School of Medicine, where I am, in the Limerick Digital Cancer Research Centre, and Mark Lawler at Queen's University Belfast. And the goal of it is to take heterogeneous cancer data and actually harmonise it into one common format, so we can actually use federated approaches to querying that data. When we say federated, that means that the data stays local. Okay. And we're using kind of common data model approach, and I'll explain a little bit more about that. Okay. Within this project, there are 7 partner clinical sites, and the goal is very much to look at sharing of genomics data, and much of that is more on the research development side, because we don't have implementation of TSO 500 panels for all patients or for large numbers of patients just yet. And then we're working at sharing blood cancer data and prostate cancer data across different sites. Okay. We are not reinventing the wheel in any of this. We are very much adhering to global standards. So one of the first things we'll do is how can we connect data, but make sure that whatever data we connect, it actually embeds itself into existing standards. Why is that important? It's important so that we can leverage European and global projects. As a small country, we can learn so much from other data. What's a very, very rare cancer in Ireland will mean that there's only 1, 2, maybe 3 or 4 families with that cancer. But when we actually look across Europe, there's probably hundreds of families with that cancer. And therefore, we can learn from each other. We can learn what worked in those countries, but only if we're all talking the same language or the same common data model. And common data models that are widely being used. So at the institutional level, people are using HL7 FHIR. So that's Health Level 7 Fast Healthcare Interoperability Resources. It's basically like a messaging system between healthcare resources. And it is a great tool. It's pretty much the standard globally now for messaging between electronic healthcare records. However, for research, this messaging system doesn't really work so well. What you actually need is you need to be able to do a longitudinal search. So you want to be able to say that I want to do a query of patients over a period of over an observation time, and then look at those outcomes. And that's not so easy to do within FHIR. The Observational Medical Outcomes Partnership, OMOP, was a common data model, or a standard for representing healthcare data that was designed by the FDA in the US and the European Medicines Agency. And what it is its approach where you actually take the data that you have and you map it to a common standard. So, for example, if I was talking about gender, in one database I might have male, female, in another I might have MF, in another it might be 1 zeros, in another could be man, woman, or there's different ways, and of course then how you might represent people that have a gender reassignment or something. Gets very, very complicated. What we would do is we could say, right, we're going to use this one standards, and everybody maps onto this one standard. And so then what we do is we map those data onto this standard, and then everything is represented in the standard format. And now what we have is we have a harmonized set of data at each institute. This has been a very popular approach, the OMOP Common Data Model. And it's being used by many, many countries around the world. Organizations such as IQVIA estimate that 2 billion healthcare records worldwide are mapped onto this format. And that's primary care, that's claims, that's secondary care, and even tertiary care. Within Europe, the largest project is, and I want to just move this out of the way, is the Darwin EU project. And within the Darwin EU project, 143 million European citizens have had their data mapped into this common data format. And this Darwin EU is a European Medicines Agency project. And this format is really being driven in large part by pharma companies who want to understand adverse effects associated with drugs. So when clinical trials are run, they're normally run for a very short period of time. And that means there's a very short observation window of the impacts of those medicines. Whereas if you have real world data or actual patient data, you can look for much longer periods of time, and you can also look at other health outcomes. So what were the comorbidities associated with patients that responded over time? What for patients that had this adverse effect, did they then, with medication 1, did they have a better outcome with medication 2? And so having real world patient data to actually do these is actually very valuable. Of course, you know, there's caveats to real world data too. These aren't completely randomized studies and that, but together they're very complementary. So I've mentioned this term federated a few times, and, you know, about security, how does it actually work? So what we actually have is we have data that's been harmonized to this common data format at each healthcare site. Now, in some cases, that's a centralized repository that a country might have or a region might have. In other cases, it is just one hospital. It could be just one GP practice. And what we have is we have data at each site that's harmonized into this common data model that is stored behind the local firewall under local GDPR and DPIA constraints. I can then query those databases. They're in the same format, so the same query can be launched at every single site. And it can go beyond queries. These can also be AI learning, where we're actually taking the weights from each of the different data sets and building AI, artificial intelligence ensemble models as well. So I send my query out to each site, but I cannot pull back any individual patient records. I can only pull back a summary or an aggregate or a weight associated with that query. Now, I can pull back a lot of information, like these were all the different adverse effects associated with this treatment, or this was the time to recurrence, or maybe the time between treatment of patients. But again, it's an aggregate, it's not individualized. And the data controller, so the individual hospital, has control as to the level of that aggregation, whether you need to have 50 patients in order to return an aggregate, or it's 5 or 10. The typical is usually 10 patients on aggregate, but maybe for a rare disease, you may need to change that, or for more common diseases, again, you may want to change that. But the local site retains control. Okay, so towards this, what I have done within the Health Hub is we've developed 2 micro-credentials. We ran the first one, BM6053, which was Introduction to Real-World Data Research in Cancer, during the summer. And this was also made available on SkillNet. So it was actually available. These are really subsidized, and I think it's 90% subsidized, to professionals. And then we'll be running a second one during the coming year. And we will probably rerun the introduction one again. The introduction is really meant for healthcare providers to talk about what data and how we use data in secondary research, and what are the standards and the things that we should be thinking about when we're actually doing research on kind of this federated approach. The second 6063 is much more designed for computer scientists who are actually writing the code and developing the methods. Okay, so data can drive cancer research, care, and policy. I think that's really, really important. In order to have robust research, we need to have studies at scale. And in order to identify those, 5% of patients that will actually respond really, really well to a treatment, we need to have scale. And that's why it's so important that we can bring together data on the island of Ireland, but also begin to integrate data in Ireland with that in the EU. For clinical cancer care, there's increasing use of these harmonized databases. So within several of the UK trusts, within Exeter, within Lancashire, and within Leeds, they're using this common data model to actually integrate over multiple hospitals in their sites. So they're getting daily dumps. The Leeds gets a daily ETL or mapping of the clinical EHR into this research format data daily. So they can do real-time surveillance and monitoring of patients. That's also being done within networks of hospitals in Texas and more broadly. Within the UK, the HDR, the Health Data Research in the UK, have said that all trusts in the UK, so all NHS data, will be mapped onto this common data format. So having that capacity in Ireland to also map our data onto this format is a really, really big opportunity. And that's really where we're starting small in Limerick, but we're hoping that we can build a much bigger network, and really by demonstrating even on a small scale that we can run pilot projects across hospitals, where essentially we're taking some of the paper records, we're digitizing them, we're converting them to the standard, and then we're actually mapping. But if we can do that kind of real-time data mapping and show the value of that, hopefully that will be a good reason to implement electronic cancer records and electronic data within the hospitals more broadly. So, and hopefully that will drive policy. Currently, there isn't enough data in a lot of cases, or the data that we have was manually pulled from the records, which means that there's a delay between the time the data was actually recorded and the actual time that those reports can be generated in order to impact policy. So hopefully we can actually provide data-driven research, or at least demonstrate one way in this might be able to be done. So there's a huge opportunity here to work together and to add benefit and hopefully provide something that's a sustainable solution. And I think this hopefully will also be able to highlight disparities and build equity in the system. For example, one of the questions that we're asking in prostate cancer is, do all patients have equal access to MRI? Which is obviously important for prostate cancer patients. Okay, and we're using standardized approaches. And with that, I kind of just want to summarize what I've talked about. So I've talked a little bit about infrastructure that we're building for cancer genomics, which is so important with increasing numbers of precision oncology and clinical actionable mutations. I talked about the importance of somatic mutations. So these are mutations that are in that we accumulate due to the environment and due to our exposures and then due to aging. And these are tissue specific, and these are observed in the cancer, and they provide a specific ways to target cancer, and that's why these are clinically actionable mutations. And understanding the patterns and the instance of those across Ireland might be really, really valuable, because it provides insights into the causes of cancer. For example, that the work from the Brennan lab, where they were seeing the specific patterns in kidney cancer in Japan, along the Danube, where there's a plant that's a kidney toxin. So we can see signatures for smoking, we can see signatures for air pollution and lung cancer. So as we begin to see these signatures and we begin to associate them with environmental causes, we can actually then develop better approaches for prevention strategies. And then finally, we also talked about clinical health records, how we're actually building minimal data set within the European Data Infrastructure Programme, and then how we're using the Odyssey OMAP system for harmonizing electronic health data, with the goal to be able to do networked cancer studies real time on the island of Ireland. And finally, I'd just like to thank various different people here. The first group here, Lauren was a master's student, so I didn't speak about her work on single cell genomics today, and Michael is also working on single cell genomics. And actually, we just had a paper accepted in GigaScience this week on approaches to demultiplexing single cell data. I haven't spoken about the single cell work that we're doing. Ahmad is another student in my lab. He's working on digitized H&E images and how we can do AI learning and associate that with genomics, which I also didn't speak about today. Most of the work that I spoke about today was from Brendan Reardon, who's a student who's based in Dana-Farber Cancer Institute in Boston in the US, but is a PhD student in my lab. He's working with Ellie Valalan and the eHealth Hub work with Mark Lawler. And with that, I would like to say thank you for listening, and I would appreciate any questions you have. Thank you. Will I stop sharing?
Yes, please. Thanks so much, Aedín. That was fantastic. I have loads of questions, but I'd like to see if I'll wait for people to maybe pop any questions in the chat, and then I can maybe call on you to turn on your camera and unmute to ask Aedín the question yourself. But I'll maybe get started. I was really interested in the information you shared at the beginning around the difference between the 2017 and the 2022 data from Memorial Sloan Kettering, with about maybe 1 in 5 patients now having a genomic target with a drug that actually has high level evidence. So I was recently up in Belfast, and one of their current selling points is that about 70% of their cancer patients are having molecular profiling. That's at least what they were pronouncing from the podium. And they obviously have an excellent biobank and molecular service up there that is driving that. So that was very interesting to hear that figure. So I suppose the question I have is, you know, and the answer is probably that we don't know how many patients in Ireland are currently, you know, getting molecular profiling, or you may know if we do, because obviously there are many genomics labs now in Ireland, and I think getting a handle on the numbers of patients being profiled, the types of mutations that are being detected is, as you said, critical for us to try and maybe attract clinical trials and match patients with clinical trials. So just your thoughts on that long comment there.
I think it's really, really vitally important that we do get this infrastructure. I think private patients are reimbursed for Foundation 1 treatment, and speaking with folks on the molecular tumor board, they're definitely seeing Foundation 1 reports. But we don't have, I think, any national database, or like even the somatic mutations, you're not sharing germline information. So as far as like CBio portal, that's somatic information. So it's normally thought to be a level lower as far as, like, you know, the sensitivity of the information. So it'd be really phenomenal. And definitely, like, I'm keen that we can set up a CBio portal instance as soon as we can, because I think it's going to be really valuable, because then we will actually have instance rates of these mutations in an Irish context, which can really drive kind of treatment decisions. But I don't know if these are being systematically collected anywhere. Within St. James's and Beaumont, I think the NCCP have upgraded their sequencers, so they have the capacity to do this, but I think it's only being done for lung cancer, for MSI, MSS, and ovarian HRD. But I don't know if that's being done for all patients, or if it was just as a trial and a pilot. I think it would be really phenomenal to be able to have it. And the other reason why I think it's important is, as the number of genetically indicated drugs increase, doing single gene testing becomes more cumbersome in labs. So if you think about it, if you have to just run one panel and it gives you like results over a lot of cancers or a lot of genes, that's easier than having to set up, you know, a multi-well plate for every single individual gene, which from a laboratory perspective, that's not actually very scalable.
Thanks. And I think just before I go to a chat question, it's just whether there's any link now between the work that you're trying to get off the ground with the eHealth Hub and the intense efforts that a lot of our cancer centres are now going to try and provide more robust cancer data as part of the European accreditation process, because that again has been an issue, is that we didn't really have good data on our numbers. And so this is now happening in a number of centres, whereby there's an attempt to have a more formalized system for capturing data in a sort of a system that doesn't have, let's say, national EHR, of course, and whether the cancer centres around the country are considering the common data models that you've referenced, etc.
So, like, we're talking to a couple of different hospitals within the project, but so within the blood cancer project, we've got one set of hospitals, and within the prostate cancer project, it's a different set of hospitals. And I think there's a couple of different things that we're doing that will be useful for that. But, you know, we only onboarded the last of the students just like last week. So we were quite delayed in getting a lot of this off the ground, because the ethics and everything else that was involved was way more complicated than I actually realized. Mm-hmm. So sometimes a little knowledge is useful, because if I think, you know, that bit of naivety let me put in the grant and actually embark on it, and then realize, whoa, everybody else was looking at me going, really, you want to try and tackle this one? So we scale back, and we definitely have like very targeted questions and projects that we're doing. So the first thing that we're doing is just that landscape analysis, which is saying, for these variables in our data dictionary, where do we find this information? Is it in structured letters? Is it in unstructured letters? Is it paper? You know, where is it? Can we find it easily in the paper record? Is it maybe in an electronic database? Is an electronic database where we've really nice mapping to SNOMED codes and things like that? And so we're tiering that data and saying exactly where we can get that in each of the different hospitals. And that's really ongoing work at the moment. And then once we've worked out the scale of the problem for each of the different cancer types that we're looking at, the next thing will then be to deliver the scripts, where we can basically do automated processing of letters to pull them into the database. And then we're already beginning to write kind of scripts that, you know, we can potentially map onto the OMAP common data model. Advantage, I think, of something like this is that we can have a local script at each site. So if you already have, you know, an electronic health record system, or you're using a system that works, we're not saying you have to map it into something else. We can just map it onto this common data model. Equally then, if we have, you know, if tomorrow an EHR system was generated, we just have to write a new ETL script. All the downstream analysis pipeline stays consistent.
Great, thank you. And I might just
any dashboarding.
Yeah, I might just shift over there. There's a question in the chat. Is there a reason for the HSE approving fewer genomic oncology indicators than private healthcare? So would you have a comment on that, Aedín? I'd have a ballpark answer to that, but I'm not involved with either process. So
no, neither am I. So the European Medicines Agency is obviously looking at the clinical efficacy and safety data, and upon that they approve a medicine. At that point in time, it's up to each individual country within Europe to decide if they will reimburse or pay for that drug. And within Ireland, HIQA have a HTA process that they undergo that looks at many different factors. So the incidence of that cancer in Ireland, and then the lifetime impact of that cancer, and the lifetime impact of that drug. So how likely is it to give a benefit, and to how many people, and then what's the cost of it? Because public healthcare, you have to work out how to best spend the money. But I think, things, hopefully some of the data, the systems that we're developing would actually facilitate that process, whereby, if we can better estimate the numbers of cancer or the genetics of those cancers going forward, then that should hopefully facilitate and expedite that process. But at the moment, the approval for some drugs can take many years.
Yeah, so part of it is around the process. The HSE or the health service have a public service process for going through carefully all of the aspects of the drug, including pharmacoeconomics, etc. And it's actually also dependent on the company putting in applications and responding to queries. So it's actually on both sides sometimes. And I think sometimes the industry partners will see Ireland as a small market, and they will actually put in their applications to the public system less quickly than they might to a large country in Europe, where they're going to get more revenue. So it's a whole process that involves multiple partners, whereas obviously the private sector maybe just makes a decision to approve something based on its cohort, which is to some degree what happens in the States, where the FDA approves the drug from an efficacy perspective, and then Medicare, Medicaid just sort of accept it as an approved treatment, and then it's immediately in guidelines and available for use. So that's the process on that side.
Yeah, I think there's definitely been a push from the pharma companies at least to say, oh, just approve it, and then you can see how much it costs over a period of time and then decide to pull it. And I think that's what's happened in a couple of European countries, is that they have a default approval where they have a limited budget for it, and then they just test it over a period of time. Whereas other countries are, there's different processes in different countries. So there's definitely different drugs available in different countries in Europe.
Exactly. So we have come almost to the end of the hour, and we're very grateful to you, Aedín, for joining us for this first lecture of our series. And the work that you're doing is absolutely amazing, and hopefully it'll transform our country for us.
That's a big ask. I don't, if we make a little contribution.
Exactly. So thanks everyone for joining, and I would just like to remind people that our next series is on November 11th, and we have 2 people joining us, and I'm just trying to, give me 1 second here. I have my notes up here. November 11th, we have Hazel O'Sullivan, one of our new consultants, who has a post across medical oncology and cancer genetics and CUH, giving us a talk on cancer genetics. So we're really excited about that. And we have Jacqueline Daly from the East Galway and Midlands Cancer Support Team, talking about the importance of community support centres in the care of our patients. So that'll be a really nice session. So we really hope that you can join us. So thanks everyone, and have a good week.